What's printing my blood vessels? My name is Jeff. I'm the co-host of How It's Med, the podcast where we chat with incredible innovators in the field of health tech, such as today's guest, Dr. Karolina Valente, CEO of VoxCell BioInnovation and assistant teaching professor at UVic. Karolina, how are you doing?
I'm doing okay. How are you doing, Jeff?
As we talked about off air, it's been a rough time, but I mean, we chatted a little bit about how busy you are with your pre-seed financing as well. Like all of that running back and forth, chatting with investors and
Mhm.
managing your company as well. That must be super busy too, eh?
It is. It's definitely a full-time job on top of everything. So fundraising has been interesting. We are in our pre-seed round right now. So 60% of the round is closed. We are seeking the remaining, and it has been a lot of conversation with investors and also a lot of conferences going on over this summer right now. So we have been going and presenting in a lot of different places, US, many times. So that has been definitely quite interesting and busy. And the team is growing. We are now almost, I think, 14 or 15 people. So the team is definitely growing too. So has been keeping me busy for sure.
Yeah, I ask this to almost everyone who comes on, but how do you balance that all? Because managing a team, flying all over everywhere to raise, pitching, all of that seems like it takes a superhuman amount of time to get this company going, keep going.
Yeah, it definitely requires a lot of dedication. I'm not gonna deny that, and requires a lot of time management. But I think the important part right now is I have a very strong management group finally. So it took some time for us to get to that point in which the management team is actually quite strong. So I have been able to delegate tasks very efficiently. So I think that took time. That took time that definitely took time to get there, and it took a little bit of resistance on my part too, but I think we finally clicked, and that has been helping a lot. Yeah.
I
Yeah,
totally understand the steps needed to be taken in that direction. It's hard sometimes,
it
but on a
is.
more human lens, like what are you doing nowadays to recharge? Like, do you even have time to recharge at all?
I mean, I always joke that I don't understand the concepts of hobbies, because I don't have much. So,
You
but
and me
the
both.
beauty, yeah. So I don't really understand the concept of hobbies. But what is interesting is I think is on my work, especially in VoxCell, I'm allowed to be myself, and I'm allowed to do what I love. So I think that's also why I don't need extra hobbies outside this, because I actually love my role at VoxCell. And on top of it, the UVic job, I actually love teaching. I love inspiring people, which is a little bit of what I do at VoxCell too. So I don't think I need extra hobbies, but it's definitely a fine balance, and it's definitely very long hours. Um, and what keeps me going, to be very, very honest, is this amazing group of people that are working with me in this company. And they are the ones that keep me going. Yeah. So,
Couldn't have spoken it better. But speaking more about, I guess, congruency with who you are and your journey, we talked a little bit like way before about how you didn't begin your journey like way back at the beginning. You didn't begin with engineering. You began with an interest in humanities. Tell me a
yeah.
little bit about how that started.
Yeah. So I think so I did my high school in Brazil, and that's you need to I believe on the last year at grade 12, you need to define where your path is going to go in terms of a university, and I really wanted to go to law. I really wanted to make a difference, and I'm quite good at speaking up. I don't hold back my opinions. I'm very good at that. So I really wanted to go to law, but I knew at that point that I was going against um a little bit of what my family background was, because everyone in my family is a mechanical engineer. Every single person in my family is a mechanical engineer except my mom. My mom is an electrical engineer. And I think mid grade 12, I changed my mind. So I decided I think I'm gonna go to engineering. And at that point, as I mentioned to you, I had already been taking a lot of history and geography that was very intensive. And I said, oh my God, now I have to catch up on all the math and physics and chemistry. And I did it, and I went to chemical engineering. So it was an interesting, because at that point, I was in Brazil. That's where I did my undergrad. And the oil and gas industry, as you can imagine, is pretty big in Brazil. And that's what I wanted. I wanted as my dream job as to be deployed in the middle of the ocean for 15 days and then come back for another 15 days. And I am not scared of working very hard. So that's what I wanted as my dream job. And then when I went to chemical engineering, as I started seeing how interdisciplinary engineering could be, and that was not something that I knew before going to engineering. And then the path started changing from the oil and gas into more the medical, the biomedical side. That's what ended up um, taking me here.
Yeah. Yeah. That I guess has that's quite the turn from an
Yeah.
interest in law to oil and
hmm
gas all the way to biomedical engineering. Did you ever manage to actually get onto one of the rigs at all?
Not really. My uncle worked his whole life like that, and I think I got to see a little bit the benefits, but I also got to see a little bit of the downsides, the impact
Mhm.
that it has on the family of being away for so long. The fact that they had to move a lot of times during the year, depending on where he was assigned to. So that part for me was not very interesting anymore. Yeah.
Yeah, yeah. As you grow up, I think the whole family aspect starts to become more apparent for sure.
Absolutely. Absolutely.
Yeah. So, I mean, you mentioned or you left off your little, I guess, snippet there about how you went into biomedical
Mhm.
engineering. Were there any particular, I guess, incidents that
Mhm.
inspired you at all to further pursue that field? Because you had to somehow go from biomedical engineering in Brazil to, I mean, the rest of your
Mhm.
journey, which we talked about, but which I don't want to spoil right now.
Yeah, no, that was my first year of undergrad in chemical engineering in Brazil. I had the opportunity to do a co-op, which doesn't happen very often because co-ops are normally second or third year in Brazil. So I had the opportunity to do a co-op. I was selected to do a co-op, and one of my first labs doing a co-op was actually working with polymer particles that are injected in the patient before they get their tumor removed. So cancer patients that have tumors that are quite vascularized, see how things ended up connecting in the end, but that tumors that are quite vascularized, they were injected polymer particles to kind of stop those blood vessels feeding the tumor, which allows the tumor to shrink a little bit. So that shrinking part of the tumor allows for removal more easily. So that was my first co-op experience, and I ended up doing two more co-ops with them in the same lab. And that was my first time seeing that engineering could be have a very big impact in the medical field. And then from there on, it was going towards my master. So I moved from Brazil to do my master's in Portugal, also in chemical engineering, but my master's was also, again, more towards biomed, in which I was studying the blood oxygenators. So the ECMO machines that in which the blood is recirculated outside the body, oxygenated and put it back when people are having heart or want surgery. And I was trying to create better membranes for those blood oxygenators to oxygenate the blood faster. So, and then from there on, I came to Canada to do my PhD. And at that point, I knew that I could define my path on the PhD was a little bit more flexible on that aspect. And I knew I wanted to do something related to cancer. So cancer was always my passion. And I never quite understood why it has been around for such a long time. And we have done some progress, but not too much progress. So I wanted to study cancer with that engineering perspective. And that's what I did when coming to Canada to do my PhD at UVic.
hmm I think that's a really good overview of your journey
hmm
overall. But I
hmm
think that was a multi-year long journey. You
It
had
was.
to, yeah, you had to go through some pretty extenuating circumstances to get to Portugal and then
hmm
to come to Canada. I guess
hmm
could you elaborate more on those difficulties that you faced? Because
Yeah.
I mean, your path wasn't a straight line from Brazil to Europe to Canada at all, and I think that's absolutely fascinating.
Yeah, and I think that's what happens with a lot of the young people at the university. Not all of them have a plan, and I didn't have a plan. I knew the passions. I knew and that's the difference. I'm good at everything, and anything I touch, I can easily understand and become good at it, and that's by working hard. So that's what it is. So I knew that any path I took, I would eventually thrive. Um, but the passion that I really had was that connection with medicine, but not enough to go to med school. That was the fine line there. Not enough to go to med school because I can be a little bit soft sometimes. I'm not very soft, but I can be a little bit soft. And then going to med school, I'm not sure if I was built to interact with people and learn their background and then engaging in a sense of learning the disease and so on. So I'm not sure if I'm good at that aspect, if that makes sense. But I didn't have a plan. So I let life take me and any opportunity that I saw, I just did what I could. So when I went to Portugal, actually the plan to go to Portugal was to do my last year of undergrad and go back home. So that's the difference with me is I make opportunities, I make things happen for myself. So when I went to Portugal and I did my 6 months, I said I'm not going back. So I'm gonna find a way to stay here. So I transferred my entire undergrad to Portugal. I transferred everything. I ended up graduating from my undergrad from Portugal. So I have no degree from Brazil actually. Even though I did 95% in Brazil. And in Portugal, I said I'm staying and I need to find a way to do my master's degree here. And then learning about this professor in Portugal that was doing these membranes for blood oxygenator, I said, "Oh, that's very interesting. I'm gonna go talk to her and I'm gonna tell her that I will work very hard." And that's what I did. And then when I was in Portugal, I met a professor from UVEC. So he was there and he was telling how great Canada was. And at that point, I thought, okay, let's try Canada for a PhD. And I applied for different places. I applied for UFT, I applied for Calgary, I applied for UVic, I applied for many different places. And then in the end I could choose where to go.
Hmm.
And that was the interesting part. The idea of coming to UVic, it was at that point, it was very strategic. I think that was the only time in my life in which I actually sat and thought, okay, now where do I go? Because I could go to UFT, it's very, very big university. I heard amazing things about that. And that point, I think was, okay, I want to be a big fish in a small pond. And that was the end of the story. So that's what it took me to UVec. I knew that if I came here, I would be able to create a large impact. Yeah.
That's I want to dig a bit more into the thinking behind that because I always have so far heard try to be the small fish in the big pond because you want to be able to learn from everyone around you. But you flip that logic on its head and we're like, okay, this is where I'll make the biggest impact. And I understand that. It makes sense to me. But like I, I, I just, I'm not able to clearly articulate as to how you could have possibly come up with that, like, counterfactual thinking.
Yeah, I think I knew, for example, and I'm gonna keep using the UFT analogy or
Mm.
comparison because it's a big university and I heard amazing things and how smart people are. And I am quite smart and I am very hardworking, but to be noticed, it takes the extra mile, right? And that's what I wanted here at UVic. And I wanted to be able to be noticed quickly. And being a big fish in a small pond, that's what allows you to do, is to be
Hmm.
able to make that difference quickly. And that was the strategy around UVic. And then when I came to do my PhD in my second year of PhD, I was already teaching the class as not as a teaching assistant anymore, but as the professor, as a sessional
Wow.
instructor on my second year. So I have been teaching here with my current role since it was my second year of my PhD. And that again, was from being able to stand out very easily in a small crowd.
Hmm. Hmm.
Mhm.
I mean, if there's anything that I've understood from talking to you from so far, you're someone who's very confident in her own abilities and her own work ethic, as well as someone who values standing out.
Yeah.
I'm not so sure that I've encountered that very often in the context of everyone that I've chatted with so far. Like, why do you think those characteristics or attributes are so important to you?
Um, I don't know. I think it's a little bit related to where I grew up. It's my family is quite big, and even Brazil, it's a massive country. To be able to stand out, you need to have that work ethic. You need to work hard. It's smart that take you so far. The remaining is working very very hard. So I think that was ingrained from my father. My father works quite hard every single day. He still does. He's 65 and still works very hard every single day. And I think I was built up like this. And then and again, it was I was born in a place that I didn't have everything. It's like even safety. It wasn't a safe place. So the whole goal for me even when I was a kid, was to find a way to get out. So I learned, I heard about this the other days, people saying, anytime you have a chance, travel in first class. Even if you don't have the money, find a way to travel in first class. Because after that, your brain is going to keep trying to find ways to go back to first class. And I think that's what happened to me when I went to Portugal, that I saw, okay, this is how life is supposed to be in terms of safety, in terms of stability, and so on. I said, I'm not going back. I'm doing everything that I can from now on to be able to provide that type of lifestyle to me. That Brazil, even though it's beautiful, it's not a safe place. So I think at that point on, I made the decision that I could work hard, and I don't get tired very often, and I just could do it. I could do it, and in my mind was, if I can do it, why not? If I have the ability, why not? So I understand when sometimes people say that opportunities were not given to them, but to be quite honest, I think to a certain extent, you create opportunities for yourself. You find out. You find a way to find out. And that's what I did. Yeah.
Fascinating. I guess like it's really cool how I guess you've articulated the fact that you've understood what it means to have a lack of safety as well as other
Yeah.
things that some people might take for given in Canada, for
Yeah.
example, and that's what drives you. But
Yeah.
I think the flip side to that as well is like, oh, as mentioned,
There you
my
go.
cat just walks into the frame.
There you go.
Um, as like how did that I guess desperation or desire
Yeah.
to
Yeah.
to keep I guess building tie into cancer? Was there anything particular that inspired you overall to go that direction? Because you've done so much work in like blood vessels, ECMO, etc. That
Yeah.
pivot to cancer doesn't necessarily click for me right away.
Yeah. Yeah. When I think the cancer it touched a little bit more on the family side, because
Hmm.
when I was younger, I remember my mom going through breast cancer, and that wasn't fun. So it touched a little bit on that
To say the
side.
least. Yeah.
Yes, yeah, exactly. And then when I had the opportunity for the PhD to do my PhD at UVic, and I even got the chance to choose what I wanted to do, I knew that I wanted to do something related to breast cancer. And at that point, I my mind was going. So we know breast cancer for a while has been around for so many years. What don't we understand yet? And then my mind started going to, well, we keep saying that cancer is very heterogeneous. There, all the cancers are quite different, but there has to be something common around them. Like it has to be something common between the different types of solid tumors. That's what I'm talking about. And that's where my mind started going. Let's try to understand from an engineering perspective, what is the impact of the cancer environment inside of the body that is making drugs not reach to them properly. And that's what became the big question of my PhD. And of course, the more you learn, the more you read, and the more you try, you can start narrowing down. And I started narrowing down to the really key um, mechanical parameters in the cancer environment that makes drug therapies become quite inefficient in terms of delivery. Yeah.
Hmm. Hmm. I guess you mentioned solid tumors there right away, and just for those who are less
Mhm.
familiar with the solid versus liquid classification of tumors, could you give like a brief, 30-second rundown uh, in a way that you would speak to a 5-year-old to explain the work that you were doing in the
Mhm.
microenvironment space? Because I think to some
Yeah.
extent that ties into and builds a basis for what VoxCell is doing to some extent.
Yeah, absolutely. Yeah. So when I'm talking about solid tumors, I'm talking about tumors that actually have a specific location inside of the body. So it's sitting inside of an organ, for example, and it's sitting inside of that healthy cell environment, inside of the happy environment in which the organs are, and it's kind of localized in that area. Of course, that it can migrate from different locations inside of the body, but it's having an impact on that healthy tissue in that healthy organ, and it's changing that environment of the healthy organ, and that's what voxel is trying, what we are investigating and trying to mimic it outside of the body.
Hmm.
Yeah.
Okay, so I'm going to talk
Mhm.
a little bit more about your PhD now because we've talked a little
Okay.
bit about what it was about that you
Yeah,
started teaching in it, but you did your PhD in microfluidics.
I did.
Um, and I guess was there any particular reason why you took that particular route? Like I can vaguely connect the idea of microfluidics to the microenvironment which
Hmm.
cancer cells have.
Yeah.
But was it like a specific moment, or did you have to spend time narrowing down, as you said, into that area? And was there any guidance by specific instructors that allow you to pursue that
Hmm.
path to your own customization?
Yeah, no, that's a very interesting question. So the microfluidics, when I started my PhD, the microfluidics was kind of, the field was kind of exploding with the new organs on a chip and people trying to mimic organs inside of this device. And what is interesting, and I'm teaching a microfluidics course right now at UVic during the summer. That's what I teach. And what is interesting is everything that you learn from fluid mechanics, everything that you learn from fluid movement in large pipes, when you scale that down, it changes a lot. But we understand that scaling laws quite well. So we understand how to mimic everything in a micro scale quite well. And the microenvironment of the microfluidic, what it allows is fluid manipulation in a sense that you can mimic blood vessels very well. And because so the way that it works, and I'm gonna just briefly explain this, it's in a large pipe. If you're passing water, when you scale that down, water behaves like honey, so it becomes quite viscous because it's so much interaction with the wall of that small channel that the viscosity of the fluid becomes a very, very important parameter at that point. So it was quite interesting to be able to mimic the blood vessel in terms of drug delivery in these small channels with having a very high control of that fluid movement. So the microfluidic was my idea. At that point, I was reading a lot about organs on a chip and people trying to mimic tumors and inside of this device. And my whole goal was actually to mimic not just the tumor area, but the drug delivery side. So the way that my device worked, I have a main channel in which my tumor was, and I had two side channels in which my blood vessels were passing, delivering drug. So I was able to mimic not just the blood vessel movement of the fluid, but also the pressurized fact that blood vessels are pressurized. So I was able to mimic that too. And another parameter of the tumor microenvironment, and going back to the solid tumors, is that you have these cancer cells growing in a place that they are not supposed to be. They're growing within healthy cells and they're not supposed to be there. So, what they start creating, they start kind of squeezing the healthy cells around. And what that does, it creates a very increased amount of pressure in that tissue. So Now, when before you were relying on your blood vessels to deliver drugs by that pressure gradient in the tumor area being pressurized, you don't have that pressure gradient anymore. So that's why delivery of therapies is so inefficient to some solid tumors. So I was able to mimic that inside of a microfluidic device to mimic that tumor area being highly pressurized. And now analyzing how the pressure in the blood vessel was not efficient anymore, was not sufficient anymore to deliver therapy. So that's where I spent a lot of my time of my PhD, and all of these delivery of those drugs were done by tagging molecules by fluorescence. So I spend a lot of time in front of this fluorescent microscope. I joke with people because the fluorescent microscope is in a basement at UVic, so it's very dark room. And it has a loud air conditioner. So I joke about spending many nights in there. And it was like a great environment because I love sleeping with a white noise. So it's like perfect for me to be there. It was like dark and had this white noise. And I spend a lot of my time in this very big confocal microscope machine that we have here at UVic. And that's where I spend doing all my PhD. So from there on, VoxCell was really the blood vessels are not being seen the way that they should, especially on the cancer environment. So what happens, and that goes back, and if I'm going too deep into the science, you please stop me, Jeff. But what happens is because that healthy cells now are being squeezed around by the cancer cells, the blood vessel structure of the tissue becomes quite disorganized. And what the cancer cells do is they start sending signals to the blood vessels to say, okay, feed me. And the blood vessels start growing in a very disorganized manner to feed the cancer environment. And I thought, okay, we are finding ways to, going back to my undergrad, finding ways to decrease that blood vessel around. But what if we could use that blood vessel as a benefit? If we understand very well how that blood vessel network works, what we could use that as a benefit to deliver drugs to that specific location. And that's exactly what my main goal with VoxCell. It's to find a way to mimic that tumor environment artificially. So I like to call the tissues that we are creating at VoxCell as artificial biopsy samples. That is a little piece of the tumor.
Hmm. And you just gave a little bit of a tease of voxel there, but I'm not gonna let I'm not gonna delve into the conversation quite yet
Mhm.
because we're still at the point in time when you were doing your PhD
Yeah.
and you were beginning to use 3D bioprinting. And that is the
Yes,
other aspect of voxel that
it
we should talk
really
about.
is. Yeah.
Yeah. So how did the 3D bioprinting thing come into your world?
Yeah, so after doing all the studies in the microfluidic devices and learning a lot of insights of the impact of the tumor microenvironment in this delivery of the drug, I thought, what if now I could actually use real tissues and try to mimic that? And I remember taking the bus, going to the BC Cancer Agency here to get those tissues, and they were real biopsy samples, and they were a pain to use. They come fixed, so which means that they come stable and they have resin around, so they cut that piece of tissue for you, and it's very, very fragile to use. It's very thin samples, and they gave me in this in dry ice, so it has to be kept cold, otherwise it melts, it makes a mess. So, and I remember thinking, oh my God, it was so hard to get that little piece of tissue that I got from the BC Cancer Agency. And I thought there has to be a way to do this artificially before we go into real human tissue. And I thought, can I try a couple bioprinting techniques? And that's on campus here. I had a lot of access to a lot of bioprinters at that point, and they were mostly extrusion-based printers. So the bioprinting field was born after the 3D printing field. And then the extrusion-based printers, they were basically an FDM, regular filament plastic printer, creating 2D layers stacked on top of each other to create a 3D structure. And then the extrusion-based printers, the resolution was so low that I thought, if I want to create something that actually mimics an artificial biopsy sample, I can't do on this. This is very, very low resolution. The resolution is around 300 microns to give an idea, where a capillary vessel, for example, can be 1 micron. So you need something that has that capability to go very, very high resolution. So then, and that's when I thought, well, if I don't have a printer for me that does what I want, I might as well create my own printer. That's where the path of my PhD. I was trying to wrap it up. My supervisor was telling me like, just stop. And people normally finish their PhDs with about maybe 1 or 2, 3 papers. Sometimes I had already 13 papers at that point. My supervisor was telling me, just please graduate, just please stop. Just
Please
you have
go
enough.
away.
You have enough. You have enough. Just please go. Move on with your life. At that point, I already had 3 offers of postdocs. I had offers from the Mayo Clinic. I had offers from Stanford, and I had offers from Washington University. So I already had postdocs lined up. And my supervisor was saying, just please like graduate, just go do something else. And then so the bioprinting was nothing in my thesis. My thesis was really heavy on microfluidics, and then the delivery of radiotherapy enhances to the microfluidics to the tumor area using microfluidics. But I got quite fascinated by bioprinting because I thought that was the perfect example of how interdisciplinary engineering can be, of how an engine like a device could have so much impact on the medical field if done properly. Um, and at that point, that's when I had the idea to create my own printer. And I thought, okay, I heard about this before about university taking IP, so I might as well read the IP policy before I do anything here. And that's where I spend some of my time doing that while writing my thesis. And that was in the middle of COVID. So that was fall 2020, summer and fall 2020. So that's when it got really bad. That Canada really shut down completely. It was summer 2020. And so I was at home writing my thesis and reading, and then working on things over the weekend in order to create my own printer. So that was the path from PhD in microfluidics to bioprinting into VoxCell.
Honestly, at this point, you're just combining so many different fields, and with 13 papers, like I agree. At that point, if I was your supervisor, I'd be like, just move on, please.
Yeah.
But you got an offer from Mayo, you got an offer from other institutions as well that anyone would be happy to take, but you're not at Mayo Clinic. Why?
No. I know, right? Yeah. Um, I graduated in August 2020, and I was actually ready to go. I sold everything that I had in Canada. I sold my car, sold everything, all my furniture, everything. And then I just got cold feet. It was about maybe 1 or 2 weeks before my flight. I had a ticket, I had a house, I had everything. And I just got cold feet. And I think it was at that point, COVID was pretty bad in the US.
Yeah.
And then I was always a brave person. And so I wonder if Canada made me a little bit soft at that point. Maybe was to work 16 hours a day every day. So not too soft, but I just got cold feet. And then at that point, I had to make a decision if I wanted to start VoxCell or not. And I remember talking to the father of one of my co-founders, and his father was telling me, like, you have to do this. And then when he said that, you have to do it. And I remember asking, why, Graham? Like, why do I have to do it? And he said, because you can. And I live it by that my entire life. If I can do it, why am I not doing it? So that kind of resonates to me that, well, I can do it and I can make a difference here. So I should do it. So VoxCell got incorporated in more or less the same time that I had defended my PhD, and I decided to stay. At that point, was a very risky move because I had no jobs in Canada. I had nothing, and I had rejected 3 very good offers from US. And I said, okay, I might as well find something to do. So I remember applying for a job at UVC, and I got the job in the biomedical engineering school at UVC, and then UVic heard and made me a better offer to stay. So December 2020, that was when I became an assistant teaching professor here at UVic. Yeah, and that doesn't
Big
happen
fish meet
very.
small pond.
Yes, and that doesn't happen very often because
Yeah.
it's very rare that you do your PhD and then become a faculty at the same school. But yeah, yeah.
Mhm.
So. Mhm.
Why do you think that they made that exception?
Um, I don't know exactly if it is an exception, if that's what it's called. It's just rare. But I have been nothing but excellence in terms of my teaching skills. That one I'm 100% sure. I remember students writing in my feedback review to the chair saying, "Make her a professor. We need her here." So I had students pledging for me, which was great. But I created new courses here at UVic while I was still a PhD student. So I create courses that are now fully accredited and part of the engineering program. I created new labs that did not exist before I came here. A lot of the engineering course didn't have any hands-on experience at all. People had never seen a big lab before. I created all of that. Yep. So I did a lot for UVic in terms of the biomedical engineering program, and I just love teaching them. Yeah.
Mhm. I mean, along the path to where you are now, you also ran into a now competitor company. I'm not going to mention
Yeah.
them. You can mention
Yeah.
them if you want. And you
Yeah.
interviewed for them.
I
Tell us
did.
about that story.
I did. That was again when I was doing my PhD and doing starting to use these different bioprinters that I had available on campus. And I encountered a local company, bioprinter, and I used their bioprinter and I said, this is not how I want. I have so many ideas on how to change this. And the resolution is not I want. But again, the piece of engineering to get to that point, that's what was okay. This is quite interesting. And I think I can change a couple of things in here. And I remember the person from that company, I believe, was I I I don't remember exactly the title of the person that came here to train us on that bioprinter. And I remember telling him, "Look, why are you guys doing this? Why don't you do that, these and that?" And then he looked at me and I said, "I'm looking for a job." So again, that's all about creating opportunities for yourself. And I told him, "Look, I'm looking for a job." So I applied and I got to the interview and I got to speak to the vice president of teaching engineering of that company. And I remember telling him, if you get me, you not just get my knowledge, because I do have the knowledge in the bioprinting, I do have the knowledge in the biomaterial side, but I also have this idea for a better printer. Um, and that was a bold move, but again, the worst answer is no. And that's it. You already have a no if you don't try. And I live by that every single day, even with investors. And I'm sure we're going to get to that point. But I remember telling him, well, I have an idea for a better printer. And that's what you get when you offer me the job. And they did offer me the job, but they also said at that point they were fundraising. And I understand exactly what that means right now, that they are really focused on accomplishing milestone. And I really respected that. But I decided to start my own company instead of merging to with them. So that where it took me to VoxCell too.
Okay. So now we come to the point where you can explain to a
Mhm.
5-year-old how
Yeah.
VoxCell works. Can
Yeah.
you do that? Yeah.
Yes, absolutely. As I said, VoxCell is creating really artificial, tiny piece of tissues. That's what we are doing. So instead of using a human tissue, we are doing that artificially. And the idea now is to use these artificial biopsy samples that I like to call, or as we call in VoxCell, our human-like tissues to test drugs. The beauty of these tissues is they actually have blood vessels. So we're able to create blood vessels. So now the same way that drugs are injected through our blood vessels. That's how the drugs are injected in our artificial tissues, in our little piece of tumor. Yeah, that's what we are doing
Hmm.
at VoxCell.
Hmm. And you mentioned that the main use case right now is in testing drugs and seeing how they permeate throughout the tissues.
Yeah.
I guess, like, what are the downstream effects of this? Who will it benefit, or like, what are the not second order or third order effects of what you'll be able to accomplish with such a tissue sample that's able to mimic the microenvironment?
Yeah, able to mimic the microenvironment in terms of mechanical and chemical properties, but also the blood vessels. It's any right now we are talking about oncology. That's our focus. We don't want to stay just in oncology, but that's our main focus because the vasculature is very, very important. So any small molecules at this point can be introduced in our tissue. That means chemotherapy. Any chemotherapy drug can be introduced, and you see the effect of that drug permeating through the tissue, as you mentioned. It's actually mimicking resistance to actually dying from the cancer cell side because of that tumor environment. We are creating different versions of our tissues that are applicable for chemotherapy, radiation therapy, and hormonal therapy. So we have different versions. And right now we are starting with breast cancer. And again, that's my background. That's where I led from my mom all the way to my PhD, all the way to VoxCell, but we want to go to lung, ovarian, prostate in a few years. And we also want to start tapping towards neural diseases later on. So that's the future for VoxCell.
Hmm. And just to make sure that we frame what VoxCell is doing within the
Hmm.
grand scheme of
Yeah,
drug development, if that's
yeah,
where you're going, which I think you are,
it is. Yeah,
then how does it fit with the current schema? Because
yeah,
for those listeners who aren't as familiar with how drugs are developed, before you
yeah,
develop a drug, you got to figure out which molecule you're going to get,
yeah.
how it's going to attach to the receptor, and then you have to test it
Hmm.
with ADME, with not
Yeah,
only in vitro, so cells in a glass
yeah,
tube, but also
yeah.
in animals,
In
and then
animal
it
models.
goes into clinical trials. So
Yeah.
where in this broad continuum does VoxCell fit?
Yeah, no, that was a great total overview. So again, going back, drugs are synthesized, and you have these different drugs, and then drugs are tested in cells directly. You deposit the drugs on the cells, and then you see the effect on the cells. And then normally now people move to animal models, which means that the drugs are then injected in mice, rabbits, and so on. And then the most promising ones go to people. The challenge is from the tests done in the lab all the way to people, the translation in oncology is less than 8%. So there is a less than 10% translation between those
It's wild.
results. It is. And then when you go to animal models, specifically, 68% of the drugs fail. When you go to people, 95% of the drugs fail in oncology. So there is a lot of flaw in this process. So what Voxer is creating is a better bridge between lab and people. And again, going back to that artificial human-like sample, what we want to be able to provide is more information in these small tumors that will be more reliably mimic what happens inside of the human body because it has those blood vessels. We are trying to replace these. We are not going to replace the monolayers because the monolayers, they are beneficial because they're quick, so you have very fast information in the beginning. But we are trying to replace everything that comes after the monolayers. People sometimes use spheroids and organoids because of the 3D environment. There's a lot of flaws with that. So we are trying to replace that, and also the initial tests done in animal models. So the animal model industry, there's a lot of impact in the environment, and there's a lot of animals used every single year. So there is a humanitarian side on that aspect. But so we are trying to replace the initial tests done in animal models by being able to mimic that very complex blood vessel architecture and decrease that gap or increase that translatable data between lab all the way to humans. So the idea is to be able to eliminate the drugs that do not work sooner instead of carrying that information to animal models and to people later on. And that's what VoxCell is doing in our tissue models.
Hmm.
Mhm.
And that I guess if I were to think about the effects of that, that would mean that we would spend less time as well
Yeah,
as less money. in
Absolutely.
developing drugs that don't end up turning out or failing down the line, right?
Absolutely. So we are trying to decrease the time of the preclinical testing by providing a more reliable platform. The preclinical testing takes about 2 to 3 years. So even if you can decrease that by 20 to 30%, you can see the effect in time. And of course, the end goal, as you said, is allowing those promising therapies to get to the market sooner and to get to the hands of people that actually need. The drug development process right now, it takes about 15 years with a 95% failure rate. That does not seem a very promising um, path. So decreasing that time and allowing the correct therapies to go to animal models and go to people, the most promising ones will allow to decrease that failure rate, and to put the therapies in the market sooner for the hands of people that actually need.
Yeah. And talking about preclinical drug development as well as those clinical trials, the savings are probably in the hundreds of millions, which means that
Exactly.
you're able to run through more drug candidates with the savings that you get as a result
Yeah.
of not going through with drug candidates that would otherwise fail.
Absolutely. And that's all the other beauty of our teaching models. They are very, very high resolution and they are small because they are very high resolution. You don't actually need a lot of sample. And when you don't need a lot of tissue in terms of size, in terms of volume, you can do high throughput screening. So you can have multiple tissues running the same drug in a very, very small quantity, multiple times. So you can have a lot of reliable information. The tissues that we are creating can even be sectioned later on for visualization of the microscope, the same way that human samples are. So accelerating that, as you said, the preclinical, it's something about 300 to 500 million, decreasing that time, let's say by 30%, by decreasing by a year. And that it is the saving that we are estimating to be something around 100 million plus.
Hmm.
Yeah.
Hmm. And in the end, who would be using the printer?
The printer right now, what VoxCell is doing is we are keeping the printer in house. So we are keeping all the IP with us, and we're going to be selling the end of this year the tissue models ready. So our idea, as I was saying, for the breast cancer that we are releasing is to have a version that is the chemotherapy version, the hormonal therapy version, and then the radiation therapy version. The radiation therapy has a little bit more interesting properties on the vasculature that is really specific for radiation, and to be selling those tissues as standard tissue models. The resolution of our printer is 500 nanometers. So it's 600 times higher than what I was saying, the average 300 micrometer extrusion printer resolution, which means that the tissue variation between the samples is very, very low. So we are not introducing more variation to the preclinical testing step. We are providing a better platform for drugs to be tested in a more human-like way through the vasculature as it happens inside of the body. So we'll be holding the printer in-house at least for this year, next year, and be selling the consumables directly on the market. And because we are creating these tissue models with our own bioinks, and I like to refer to the bioinks as a printer's cartridge. The same way that you go to Walmart and buy a cartridge for your printer at home, that's what our bioinks are. And we are making our bioinks to be universal. And what I mean by that is compatible with any other bioprinter, independent on the type, whether it's a laser-based or not. We are making our bioinks compatible with any other printer. So we are going to be putting these bioinks on the market in August this year. So the idea is now going from the tissue models, which we are targeting to really be CROs, pharma, contract research organizations, and pharma. We are also going with the bioinks to be more of academia and research centers, people that already have their bioprinter and are just looking for a better ink that in our case mimics the cancer environment.
Hmm. So just listening to all of this between bioinks, bioprinters, fundraising, managing a team,
Mhm.
teaching, like learning how to operate a company while maintaining and coming from an academic background is difficult. The skill sets don't
Yeah.
overlap as much as I'd like. So how have you managed to develop those skill sets and how have you managed to choose the right mentors to guide you on your business development journey, given the fact that you've been thrown into it and you've had to do
Yeah.
this?
Yeah, that's a very interesting question. I remember last year sitting with my former PhD supervisor, and asking him, because when I was doing my PhD with him, he had almost like a 20 people group, because it was a large group, so more or less the same size as VoxCell right now, and asking him how he managed to manage the science, but also the different personalities, the different people that they had. And they said, Carol, this is what people don't teach you. You learn with practice. People don't teach you when you're doing your PhD how to manage people. They teach you how to do the science. And then for me, that was a high learning curve. So I got a lot of mentors from when I did the I2I program with SFU. And I also did other entrepreneurships programs here from UVic. And I started learning all that soft skills that are also required when learning, when creating a business. The science I know very well. I learned the business side quickly enough. And of course, I still need some help on that. I don't have a business background, but I learned it quickly enough. And that soft skills of leading instead of just managing. And I think that's the most important part is I keep saying to people that I lead, I do not manage. I have people to manage for me, but my whole goal is to make sure that people are happy in this environment and they are working quite hard to make VoxCell a successful company with having that goal in mind of accelerating drugs, but being also inspired by the whole mission and the vision that we have in the company. So learning how to balance that people's skills, I think that for me was the biggest learning curve last year. Yeah. I
Hmm.
thought there was one size fits all, and it's definitely not. So I started learning that people have different personalities and that we need to have different approaches. And so it has been quite rewarding for me to see. Just out of curiosity, a lot of our people at VoxCell, they all have been most of them have been my students in the past. So that was something that for me was quite easy to collect the appropriate talent, to select the appropriate talents to put in this company. And for most of them, this is actually their first job, their first real job. So it has been very rewarding. Beyond the business, it has been very rewarding to see these individuals becoming the successful people that they now are, learning the business skills, learning from this first job that they are having. And we are working at a very fast pace, and I'm very proud of all of them.
Hmm. Hmm. Yeah, no, I was going to ask you that next. Like, what is it like working alongside your students? Because that's a pretty unique circumstance to face overall. Like, I, I'm I believe you do have external hires as well,
We
and I
do.
guess my question to you would be like, what is the contrast between working with your previous students versus working with the external hire?
Um, I think everyone that works at VoxCell, they really see my vision, and I keep it joking with people saying I can teach the science. I'm not worried about people joining us and not having the science background. I cannot teach personality. So if people have the right personality and they work hard and they understand the mission, the vision, the science, I can teach. For my students that already have multiple courses with me, of course, they grab the science much quicker because they already know my teaching skills and so on. And for the new hires, took a little bit more time to get used to the science part, but with the right personality. And I think that's what this amazing group have right now. It's that common personality, that driving force. I think it's all clicking very, very well.
Hmm. So, just as a last thing to close this off, if there is one way that listeners who have exposure to work in drug development and research relevant to Voxel could help to move the platform and the company forward, what would it be?
I don't think I have one wish. I have multiple wishes, so I'm gonna wish for everything right now because you gave me the room to do that. So VoxCell right now is looking, of course, for partnerships. We are looking to pharmaceutical companies, research centers, academic people that want to partner with us and try our tissue models. As I was saying in the beginning, we are also fundraising right now, our pre-seed round, so we still have around 30 to 40% of the round to close. We did our first closing in April, so we are looking for the remaining right now. And we are always looking for talented people. So look us up at voxcellbio.com. And we are always looking for new and talented people to join us.
You took my line. I was supposed to say voxcellbio.com, but
Oh,
that's okay.
oh,
You
you
did my
can say
job for
it
me.
again. Right?
And you can find Voxcell at voxcellbio.com, and you can find How It's Med at How It's Med or howitsmed.com. Thank you, Karolina, for coming on.
Yeah, thank you, Jeff, for having me.